All right, welcome everybody to lecture number 5, which is protein structure part 2. So this is the second of our lecture series entirely on protein structure. So today, instead of primary and secondary protein structure, we'll be talking about tertiary and quaternary protein structure. So tertiary protein structure is where those secondary structure elements such as alpha helices and beta sheets will fold, and the entire protein polypeptide chain will fold into a tight, compact globular shape that will dictate its function. So we'll discuss concepts like the hydrophobic effect, disulfide bonds, motifs, and protein domains. Then we'll move on to protein quaternary structure, where fully folded polypeptide chains can interact with each other to make a fully functional quaternary protein. And so we'll also discuss principles such as protein folding, protein denaturation, intrinsically disordered proteins, protein misfolding, and chaperones, which are proteins that help proteins fold into their proper conformation. So I hope you all enjoy the lecture, and let's launch into it. Lecture number 5, Protein Structure, Part 2. All right, so before we get started today, I'll go over again the 4 levels of protein structure, which we also talked about in the previous lecture. So there are 4 levels of protein structure: primary, secondary, tertiary, and quaternary structure. So we reviewed last time primary and secondary structure. So primary structure is the sequence of amino acid residues in a protein or a polypeptide chain linked by peptide bonds. And so that primary structure ultimately dictates the overall protein fold and structure. Secondary structure is the local folding of that polypeptide chain into regular structures, alpha helices, beta sheets, turns, and loops, and that's dictated by hydrogen bonding on the protein backbone. So today we'll talk about tertiary structure, which is the globular folding of a protein into a compact structure. So this is the folding of multiple secondary structures in the protein, and this is going to be more dictated by the side chains. So hydrophobic side chains are going to be the interior, hydrophilic side chains on the exterior, and also stuff like disulfide bonds and salt bridges and just interactions between different side chains. That will make the tertiary structure. And then we'll finish out by talk about quaternary structure, which is multiple polypeptide chains interacting to form a complete assembled protein. And so this would be caused by protein-protein interactions between identical or different polypeptide chains. So here's a little bit better of a graphical illustration of those 4 levels of protein structure. So you can see the primary structure here, which is just the amino acid sequence of the polypeptide chain. So in this case, it's proline, lysine, valine, lysine, alanine, histidine, so on and so forth. Now that primary structure is then going to fold into secondary structure, such as this alpha helix here, which is caused by hydrogen bonding on the backbone of the polypeptide chain, with the side chains sticking out from the alpha helix, which is a really good representation of that here. So the side chains are shown in yellow, and that backbone is shown in blue. And then the graphical depiction of the alpha helix as a ribbon is shown in green. So we also have tertiary structure, which is where those that secondary structure is going to fold into an overall globular 3-dimensional fold of the protein, as you can see here. So this protein's made up entirely of alpha helices with these turns and loops, and then there's also a cofactor, so a heme molecule shown in red here in this protein. And then quaternary structure would be multiple polypeptides coming together to form a final assembled functional protein. So this subunit here in green, the tertiary structure, is then assembled with an identical subunit and 2 different subunits shown in pink to make the final functional protein, which in this case would be hemoglobin. So those are the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. And today we'll talk about tertiary and quaternary structure. So, as I said, tertiary protein structure is the compact three-dimensional folding of an entire polypeptide chain. So it's the local folding of the secondary structure, such as alpha helices and beta sheets, but now the whole polypeptide chain is folding based on interactions between the side chains. So this is caused by non-covalent interactions between R groups and also disulfide bonds, which are actually covalent interactions between R groups or side chains on the protein. So remember that the secondary structure is caused by hydrogen bonding from the main chain. Tertiary protein structure is going to be caused by interactions between the side chains or the R groups that stick out from that secondary structure. So this often results in very close interaction in three-dimensional space of residues that are very distant in the primary sequence. So you can see here is likely the either the N-terminus or the C-terminus of the protein, and that's interacting with a region that's close to also the N-terminus or the C-terminus. So these residues are very far apart in the primary sequence, but may have close interactions when the protein has its three-dimensional fold. Now, one of the most simple features that actually causes a protein to fold in a distinct way in three-dimensional space is the hydrophobic effect, which we talked about in the very first lecture of this course. So the folding of water-soluble proteins are going to be dictated by this hydrophobic effect, where hydrophobic side chains are going to be buried on the inside of the protein, whereas polar or charged, so hydrophilic side chains, are going to tend to orient more towards the outside of the protein. So here's a three-dimensional depiction of myoglobin, and you can see the hydrophilic residues in blue are more located on the outside of the protein, whereas the yellow residues are actually buried in the interior. And that's due to the hydrophobic effect, where hydrophobic molecules tend to orient away from water in an entropy-driven process. So this is very important for one of the most basic features of how proteins fold into a specific structure. So that would be considered their tertiary protein structure, and the hydrophobic effect is a very basic principle of that. So for water-soluble proteins, hydrophilic residues are going to be oriented towards the outside of the protein, so interacting with water, and hydrophobic side chains are going to become buried on the inside. And so that's going to dictate a lot of what you see through three-dimensional protein folding. Now, importantly, this is also going to be pretty much the opposite for certain membrane-bound proteins. So in a membrane-bound protein, such as this one depicted here, so if you think of the membrane sort of located on these portions here, you can see that the actually hydrophobic side chains are going to be on the outside and exposed within the membrane. Whereas if the protein has a channel in the middle that interacts with water, the polar charged or hydrophilic side chains are going to be oriented towards the inside of that channel. So this is a protein that's embedded in the membrane and forms this channel, and you're going to see sort of the opposite effect for its tertiary protein folding. But it's still based on the hydrophobic effect because these hydrophobic side chains are going to be interacting with the hydrophobic interior of the lipid bilayer cell membrane, and then the polar side chains are going to be sticking out in the solvent-exposed area and also in the middle in this channel. So this distinction between hydrophobic and hydrophilic side chains or R groups are very important for the overall folding of a protein in three dimensions. Now, another important feature of tertiary protein structure are disulfide bonds. So these are actually covalent interactions that form between the thiol groups of cysteine residues that are in proximity on the polypeptide chain. So here you can see we have two cysteine amino acids with these thiol groups here. So there will be an oxidation reaction with those two cysteines that are in close proximity to each other that will actually cause the formation of a covalent disulfide bond between these two sulfur atoms. And this is a very common part of overall tertiary structure and overall protein folding in general. So a good example of this is insulin, which is actually made of just generally pretty short polypeptide chains that are linked by disulfide bonds. And this linkage between disulfide bonds can happen on the same polypeptide chain, or in the case of insulin and other proteins, on different polypeptide chains. So disulfide bonds are very important and very common in folded proteins and are a good, important feature of tertiary protein structure. So now if you're looking at overall protein structure, one thing that you sort of keep an eye out are for motifs. So motifs are common three-dimensional structures that are shared between evolutionarily unrelated proteins. So these motifs can be found in very, very different proteins that have very different functions. They can catalyze different reactions, be completely different proteins, but they have these common motifs, which are just based on interactions between these secondary structures in these proteins. Although they do often have similar functional effects, including like there's specific motifs that are often involved in things like DNA binding. So you will find them in different proteins that have the same effect or even different effects just because they're thermodynamically stable sort of conformations. So we have a few different examples here. And they're pretty much named pretty easily just based on what what is there. So a helix turn helix is very simple. It's just a helix, a turn, and then another helix. And so that's very common in different proteins. So you can see the cartoon description sort of shown here. So there's a helix, a turn, and then another helix. And here you can see this in an actual three-dimensional folding of a protein. So here's a helix, there's a turn, and then there's another helix. Another one would be a beta-alpha-beta unit. So this would be two beta strands that form a beta sheet that interact with an alpha helix. So you can see that in this protein here. So there's a beta strand. It links up to an alpha helix and then to another beta strand that interacts. And then all three of them interact as a little unit or a motif. Another one would be a beta hairpin. And so these are very common. So just a beta sheet and then a hairpin turn and then another beta sheet, and these interact with each other. So you'll find these over and over again in different proteins as part of their overall tertiary structure. So here's a few more examples which have a little bit more fun names. So one would be a beta meander. And so you can see that that's sort of where a beta sheet sort of keeps making these beta hairpin turns. So there's a beta sheet going in this direction, and then a turn, beta sheet in the other direction, turn, and so on and so forth, just like a sort of winding river that sort of meanders or goes in different directions. And you can see an example of this type of motif in this protein right here. Another one would be a Greek key, which is sort of based on this sort of interesting something like that, that you would find in art in old ancient Greek stuff. So that would be like a beta sheet starting here, interacting with another beta sheet that interacts with another beta sheet, and then interacts with this other beta sheet on the other side. So that would be a Greek key motif, and there's something similar shown in this protein right here. And then, which we saw in the last lecture, actually like a beta sandwich. So these are beta strands or beta sheets that actually interact one on top of each other. So you have these beta sheets, beta strands that are interacting with each other to form a beta sheet, and they sort of form a sandwich with another beta sheet behind them. So this protein right here is basically one big beta sandwich. So these motifs are very, very common, and you'll run into them in different proteins and are an important part of tertiary protein structure folding. And so another important part of tertiary protein structure are domains. So protein domains are compact regions of a polypeptide chain that fold independently from one another and often have a conserved function. So they will maybe catalyze a specific reaction or bind to a specific molecule. So here's an example here. I believe it's an immunoglobulin domain. So this is going to be involved in binding to other proteins. And they have these 4 different domains that are similar in structure. They're made out of beta sheets, but they fold independently from one another, but they have a conserved function. So protein domains are these compact regions within a full polypeptide chain that fold independently and often have a specific conserved function. And so I have an example of how these protein domains function to actually influence the overall activity of a protein as well and are conserved among many different proteins. So here I have an example of 3 different phosphatases. And so we'll discuss these later in the course. So these are proteins that remove phosphate groups from molecules. And so in each of these proteins, they have a conserved catalytic site. So this is actually the catalytic site right here, which is shown in red. And each of these proteins catalyze the same reaction, which is the removal of a phosphate group from another molecule. And these catalytic sites are located within this phosphatase domain. So it looks a little different on this one. I think there's an extra domain, but all 3 of these proteins have a conserved phosphatase domain. So these are the phosphatase domains. And so each of these proteins do the same thing. They have a very similar fold in this domain that folds independently from the rest of the protein and has that catalytic site, and they all remove phosphate groups from different molecules. Now, you'll also notice that there's an additional domain on each of these proteins located right next to the phosphatase domain. So in this protein here, this is a protein binding domain, whereas in this protein here, which is called P10, this is a lipid binding domain. And in this protein here, which is called starch XS4, one of my favorite proteins ever, this is a carbohydrate binding domain. So now you can sort of get a better idea of how domains work. So all 3 of these molecules are able to dephosphorylate or remove a phosphate group from different molecules, but each of them have an extra domain which will dictate what kind of molecules they remove phosphates from. So here on the left, I think this is PTP1B. This will remove phosphate groups from different proteins. This one in the middle, P10, will remove phosphate groups from lipid molecules on the membrane, the lipid bilayer of the cell membrane. And then over here we have starch XS4, which will actually remove phosphate group from starch molecules, which is a carbohydrate. So they each have these conserved domains, but then they have these different domains up here that can actually dictate the specific activity of each of their proteins. So I hope that gives you a little bit better idea on how tertiary protein structure, especially these domains, can actually dictate, and the 3-dimensional folding overall of the protein can dictate how the protein functions. All right, so now that we have a handle on tertiary protein structure, we can move on to quaternary protein structure, which is where multiple polypeptide chains that are all folded into a tertiary structure can combine to form a single functional protein. So these would be called oligomeric. So oligo means more than one, and then meric, so there's multiple what are called subunits in these proteins with quaternary structure. So each of the individual polypeptides are called subunits, and they're held together by many weak non-covalent interactions. So hydrophobic interactions. So maybe there's a hydrophobic region between in each of these different subunits that causes them to interact. Electrostatic interactions, so between positively charged side chains and negatively charged side chains, and then also hydrogen bonding interactions between the proteins. So these are all weak, non-covalent interactions, but when there's a lot of surface area between these proteins, it can make for a very strong and stable quaternary structure in the protein. So importantly, a lot of the proteins, like the ones I just talked about in the last slide, do not have quaternary structure. So proteins without these multiple subunits do not have any quaternary structure. But proteins like this protein, which is hemoglobin, does have quaternary structure because it has these two identical subunits and these two identical subunits that all interact and are all necessary for that interaction to have the full function, full and proper function of the protein. And so the quaternary structure, so the interaction of these different polypeptide chains can happen between two of the same polypeptides or two different polypeptides. And they can also happen in a lot of different numbers within these oligomers. And there's sort of naming conventions that go along with that I would like you to know. So yeah, subunits in oligomeric proteins can be identical or different polypeptides. So the assembly of two identical subunits, which is an example here, this crow protein from the bacteriophage lambda, that would be called a homodimer. So homo for the same, and then di for two, and then mer for the oligomers. So this would be a homodimer. If they were two different subunits, they would be a heterodimer. And so here's an example that's been very important in everybody's life over the last couple of years. So this is a picture of the spike protein from SARS coronavirus 2. And the spike protein is actually a homotrimer. So it's an assembly of three identical subunits. You can see here in blue, orange, and turquoise. So if there were three different subunits, it would be called a heterotrimer. But because the spike protein has these three of the same polypeptide chains that interact on the virus surface, the spike protein from SARS-CoV-2 coronavirus 2 is a homotrimer. And so the example that we looked at earlier of human hemoglobin, so human hemoglobin is made out of two alpha subunits and two beta subunits, and so human hemoglobin would be a heterotetramer, so tetra for four and mer for, multimer. So if there were four of the same identical subunits, it would be a homotetramer. And those naming conventions go on and on, the more subunits that are in your protein. So now that we've gone through how proteins actually fold in 3 dimensions, let's talk about a little bit about how proteins can become unfolded. So unfolding of proteins is called denaturation. So that would be the loss of the native structure of a protein, which would result in the loss of its activity, because a protein obviously needs to be properly and fully folded for it to have activity. So this can be achieved by increasing temperatures or the addition of chemicals such as urea, which is shown here, which is a common chemical that's used in the laboratory to denature proteins, to cause them to not be folded properly anymore. And then the increased temperature, you do that all the time. So if you cook an egg, the conversion of the clear runny parts of an egg to the opaque white parts of an egg, that's basically a denaturation process where the proteins, the albumins in that egg, become unfolded. And disulfide bonds can also be broken in the laboratory by the addition of reducing agents, such as beta-mercaptoethanol. So because these are covalent disulfide bonds, you need a little bit extra chemical process to get rid of those covalent bonds. So beta-mercaptoethanol would be a reducing agent, so it would reduce this disulfide bond and cause it to break. and both of those cysteine molecules are now protonated. So this is denaturation, is the unfolding of a protein, either by increased temperature or the addition of chemicals such as urea. And then you can break disulfide bond by the addition of reducing agents. And so experiments in the 50s by this scientist Christian Anfinsen were actually very important in showing how the primary sequence of a protein and its its overall tertiary or quaternary folding causes the protein. The protein requires that folding for it to have activity, and that the primary sequence of a protein also contains all the necessary information to direct how that protein is going to fold in 3 dimensions. So we'll go through that experiment very quickly. So the first step of the experiment that they did was to take a protein called a ribonuclease, and they purified it with its native structure intact. So it was a purified, properly folded, in 3 dimensions ribonuclease, and it was catalytically active because it's properly folded. Now, if you take 8 molar urea, which would cause it to become denatured, and beta-mercaptoethanol, which would cause the disulfide bonds to be reduced, now you have a denatured protein that's unfolded, and now this ribonuclease actually has no activity. Now, the interesting part about this experiment, and it adds a little bit more of a layer of just folding or unfolding, when that urea and beta-mercaptoethanol were removed from that solution that contained the denatured ribonuclease, the protein actually gained back its function. So the protein was able to fold back into 3 dimensions again and gain back its function. So this not only tells us that a protein has to be properly folded for it to have its proper activity, but it also shows us that all the information on how that protein folds into its proper conformation is contained in the primary sequence. So once it was denatured, all you had left was a linear polypeptide chain with a very specific amino acid sequence. And when you removed that urea and beta-mercaptoethanol, you didn't have to do anything else to that protein. It just folded back into its regular conformation on its own. So that's actually if you think about it, a very surprising sort of result. So at that time, you would have thought maybe, the protein has to be folded right while it's synthesized. You can't unfold it and refold it. Turns out you can, and the only information that you need for it to quote unquote know how to fold into its proper orientation is that primary protein structure. So this is a good demonstration of how the primary protein structure dictates the overall 3-dimensional fold of the protein. So please understand this experiment. It's a very important experiment when you're talking about protein structures. So please understand this experiment and what the ramifications of it are. And so this is a little bit confusing, really. So how do proteins, quote unquote, again, know how to fold into their proper orientation? So there's a concept called Levinthal's paradox, where proteins fold much faster than they theoretically would if they had to try every possible orientation. So you can imagine when people were first thinking about how proteins folded, they'd be like, well, there's 200 amino acids or 500 amino acids. Maybe the protein just quickly adopts every possible conformation it could, and then the most thermodynamically stable conformation wins out. Turns out proteins, that would take way too long, and proteins fold much faster than that. So subsequent work has found that proteins fold by sort of retaining partially correct intermediates. So they'll sort of be, and this sort of funnel here is a good depiction of that. So they'll find one part of the protein, and this image here, so one part of the protein will sort of fold pretty quickly and find a thermodynamically stable sort of conformation, and then other parts of the protein can fold as well until you get to a fully folded, thermodynamically stable conformation. So you can kind of think of this like the game that's popular right now, Wordle. So Wordle would be way more difficult if it didn't tell you any right or wrong letters in this 5-letter combination. So so they were able to figure this out in 6 guesses, basically because it told them that, oh, yeah, this RA part is properly, if you think about it like a protein, is properly folded in the right conformation. And so the protein can sort of try other conformations built upon that. And so proteins fold by what's called intermediate steps. So local areas will become folded, and then the rest of the protein will sort of be built on those correct conformational foldings. So we've talked a lot about proper protein folding and how a protein has to be folded in the right conformation for it to have its function. And we've shown a lot of images of membrane and globular proteins that are very, discretely folded into very specific conformations. But I do need to note that there are some proteins that are called intrinsically disordered proteins. And so they have functions in your cell, but they lack a defined, fixed, three-dimensional structure and are dynamic in solution. So here is a sort of depiction, which I believe was determined via NMR, which we'll talk about later, of a protein. And you can see it sort of adopts lots of different conformations. And what you don't see in that protein is a lot of beta sheets or alpha helices or any other secondary structure that would lead to an overall binding via tertiary structure. And that's because this is an intrinsically disordered protein. It's sort of, its structure causes it to be disordered, and that's related to its activity. Now, also importantly, often these intrinsically disordered proteins do adopt a more fixed conformation or folding upon interactions with other proteins. So, for example, there would be this intrinsically disordered protein that's sort of flopping around in your cell, but when it interacts with another protein whose function it influences, it adopts a more alpha helical sort of conformation. So intrinsically disordered proteins, they are out there, they do exist, and they often have very important functions in your cell, and they lack fixed conformations, sometimes for their whole lives, but also until sometimes they interact with a different protein. And so we've also talked a lot about proteins folding properly, but there's also protein misfolding is a real problem with a lot of different diseases. So protein misfolding can actually result in a lot of different diseases stemming from the aggregation of these misfolded proteins. So Alzheimer's disease, Parkinson's disease, and Huntington's disease are all terrible, terrible diseases, and they actually result from these accumulations of misfolded proteins in your brain. And you can sort of see the ramifications of that from this brain scan here on the left. And another interesting feature of protein misfolding are prions. So prions are very interesting, actually. They're misfolded proteins that have the ability to induce misfolding and aggregation of these proteins in properly folded proteins from the same gene. So that may sound a little confusing, but basically you have these proteins that are in your body right now. They're properly folded, but if you introduce the same protein that's a bit misfolded, it can actually cause misfolding in those properly folded proteins and cause these same aggregations in your brain as compared to the same type of aggregations compared to Alzheimer's, Parkinson's, and in Huntington's disease. So prions are actually considered sort of an infectious agent, even though they're not bacteria, virus, fungus, anything like that. They're just proteins. And actually the mad cow disease in cows, Creutzfeldt-Jakob disease in humans, scrapie in sheep, and chronic wasting disease in deer are all caused by these prion proteins that are misfolded and cause other proteins in your body to misfold as well. So when prions were first discovered, a lot of people did not believe it at all because all infectious agents must be, a bacteria, a virus, fungus, or a parasite. but they turned out to be correct. And these are actually just proteins that are misfolded and can cause disease in humans. So protein misfolding is very important for disease manifestations. And so how does our body do so well at folding all of our proteins in the proper conformation? And thus, we're not all have Alzheimer's at in our 20s or 30s because of these misfolded proteins that accumulate. Well, we talked about the thermodynamic stability of proteins and how they fold in a thermodynamically stable conformation. But also, there's other proteins that help facilitate the proper folding of proteins. And these proteins are called chaperones. So they're named sort of like, a chaperone that would take you to the dance. These are chaperones that help other proteins fold into their proper orientation. So I just wanted to mention them because, it is scary. There's diseases that happen from protein misfolding, but this doesn't happen all the time in your body. Proteins do misfold, but they can sort of be taken care of or disposed of by your cell. But chaperones are proteins that actually do help proteins fold into a proper conformation. We don't have time to talk about exactly how they do that, but they are interesting proteins in their own right. All right, that concludes lecture number 5, which is Protein Structure Part 2, where we discuss the tertiary and quaternary structure of proteins. So there's a few points that I really want to emphasize here at the end. One is that you have to understand what tertiary protein structure is. So it's proteins folding into a compact shape that dictates their function. So that secondary structure we discussed before, alpha helices and beta sheets, will actually interact with each other based on the side chains of the amino acids, and the protein will fold into a compact structure. And we also discussed concepts such as hydrophobic effect, motifs, protein domains, and disulfide bonds. So you'll have to know what each of those are. We also discussed quaternary protein structure, which is how those fully folded polypeptide chains will interact to form a complete protein. So not all proteins have quaternary structure, but many of them do and require specific quaternary structure for their function. And we also discussed additional concepts such as protein folding in general, denaturation of proteins. So you'll have to remember the Anfinsen experiment, intrinsically disordered proteins, diseases that can be caused by protein misfolding, and also you need to know what a chaperone is. That it's a protein that helps other proteins fold into their proper orientation. So I hope you all enjoyed the lecture, and as always, please let me know if you have any questions.